Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, Eliminating Bottlenecks and Managing Quantity and Quality in Genomics Research. It's widely accepted that the future of healthcare lies in personalized medicine. In the not-too-distant future, the type of care you receive and the kinds of medicine that will be prescribed for you will depend on your genetic makeup. Driving this healthcare revolution is the field of genomics. Today's webinar will focus on showing you how to automate your own genomic workflows to improve productivity and efficiency. Also to be explored will be how to overcome any upstream or downstream obstacles that may occur as you design and implement your automated genomic workflow systems. We've assembled an expert panel on genomics, so we'll meet them in a minute, but I want to introduce them. Louis Murray is Applications Market Development Manager at Agilent Technologies. Louis will waste no time in getting right to the point of today's webinar. His talk will go straight to the topic of eliminating bottlenecks in genomics workflows. Daniel Tessier serves as Vice President, Technology Centers at Genome Quebec and at the McGill University and Genome Quebec Innovation Center. Dr. Alexander Montpetit is the Assistant Scientific Director at these same institutions. Daniel and Alexander are going to make a joint presentation on how they address the issues of genomics workflows and tackling bottlenecks. They will also offer some tips on how you can improve your own operations in this area. I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotechnology News, and I'm going to serve as moderator for today's webinar program. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console, and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So if everyone's ready, let's get going. Louis Murray will be our lead-off speaker. Louis, the floor is yours.
Thank you very much. Hello, everyone. Welcome to the webinar. My name is Louis Murray, and I'm the Applications Market Development Manager for Agilent Automation Solutions. And today, I'll be speaking about the elimination and moving of bottlenecks in genomics workflows. So first off, I would like to start by talking and commenting on some common thoughts about automation in the lab. So why automate in the lab? First off, to increase throughput. This is how many samples can be processed in a certain amount of time. We can increase walk-away time. So this is the time between sample loading and unloading, time that someone is now free to do something else while the automation runs part of the process. Thirdly, we can increase the data reproducibility. This reduces the day-to-day or person-to-person variability in sample handling. And most importantly, this allows us to make better comparisons of our data over time and from sample to sample. So both points 1 and 2 in this slide remove bottlenecks in a workflow. Now, first off, I'd like to explain what a bottleneck is. So as an example of a bottleneck would be if a lab has an instrument capacity to run, say, 4 plates on a sequencer in one day. However, they have the capacity to prepare sequencing plates at a rate of 12 plates every day. In this case, the bottleneck is the sequencers. So the analogy is that we have a wider bottle full of ready plates that are going through a narrower neck of the bottle, in this case being the sequencers. So here we'd really need to increase our throughput on the sequencers threefold in order to address that bottleneck. So as I mentioned, both 1 and 2 and the points of this slide remove bottlenecks from our workflow. Or do they? So now, what I would like here is to consider the moving versus the removing of bottlenecks. So automation will remove one bottleneck and increase the throughput of a process. However, in reality, it simply moves the bottleneck to another step in the overall process. By increasing capacity in one area, we then put more load onto another area. So when trying to increase a sample processing capacity and planning the growth of automation in our lab, it is key to firstly identify where the bottlenecks are, where they would move to if addressed, and how much additional capacity is enabled by addressing different parts of the workflow. So by carefully planning and coordinating lab expansion efforts, throughput can be increased as efficiently as possible, ensuring that the different processes grow in synchronization. By identifying where the bottleneck in a process will be moved, a lab can plan ahead more effectively in growing its automation capabilities. So now I'll give an example of moving a bottleneck in a genomics workflow towards the nucleic acid normalization. So first off, starting upstream in a process that requires normalized samples. So one of the initial bottlenecks which greatly benefits from automation in a next-gen sequencing workflow is the library prep portion. This is really a labor-intensive process involving complex pipetting and many short incubations, which can really be addressed very successfully by liquid handling robots and scheduling software. This is typically one of the first bottlenecks or pain points for a lab doing NGS sample prep in larger numbers. So as our guest speakers will be discussing later, the Agilent Bravo liquid handler has a very successful microplate-based solution for processing anywhere from 8 to 96 samples in a run. Provides a compact and open platform which can scale with NGS sample prep needs. So in the table on this slide, I'm showing some of the throughput benefits of automating one of many possible reagent kits on the Bravo. In this example, the SureSelect XT chemistry. The pink columns in the table show the hands-on time of an experienced user prepping samples in tubes in a staggered manner for maximum efficiency. Highlighted by the red circle, we found that one individual in a lab can process up to 20 to 40 samples per week. So comparing that with the columns in green, where I'm showing the throughput of a Bravo running an automated method, we greatly increase the throughput up to 192 samples per week, effectively running 296-well plates. So here we not only include the throughput, we also streamline the process, we reduce the hands-on time, minimize user error, and improve reproducibility. These are all the combined benefits of automating a process in the lab. So with that bottleneck addressed, I'd now like to look at the overall workflow to identify adjacent processes where the bottleneck could now have been moved to. So from this workflow, we can see here that biological samples are submitted, the nucleic acid is extracted, samples are normalized, and then used for the input into library enrichment process. Samples are then pooled and then sequenced. So depending on the equipment already possessed by a lab, the bottleneck could be moved to any one of these areas. In my example, I'd like to show how to address the normalization as a common next bottleneck to be addressed. So starting samples normalized to a uniform concentration are required for many of the next-gen sequencing library prep chemistries. And each chemistry requires its own particular starting concentration. For example, shown here, Haloplex requires 45 microliters of sample at 5 nanograms per microliter, while SureSelect requires 130 microliters at 23 nanograms per microliter. So if a lab now needs to normalize hundreds of samples per week, this process can really benefit from being automated. So what's involved in the normalization process? First off, we start with unknown samples of unknown concentrations, which must be assessed. Then appropriate dilutions must be made with a diluent. And once diluted, the final concentration must be confirmed. So now I'm going to explain how this new bottleneck can be addressed by looking more closely at the individual steps of the process in my next slide. So here I break down the normalization workflow into more detail, and I can start to see what functionality or capabilities are required by an automated solution for each of these steps. So breaking this down, in the concentration determination, I'm starting with transferring of liquids from tubes to plates, doing plate-to-plate transfers, and also detecting the reagent output on a plate reader. And then creating a dilution plate, once again, doing liquid transfers from one plate to another, and also bulk transfers of reagents. Finally, adding the stock nucleic acid to make the appropriate dilution in the plate. I must once again do liquid transfers from tube to plate. and very accurate low-volume pipetting is required. And then in the last step, the post-normalization concentration determination, I'm once again doing liquid transfers from plate to plate, and then using a plate reader to determine the signal and the concentration. So if I look at this same diagram here and highlight in different colors the common requirements for the normalization process, and the most common steps here are highlighted now in red, and these all correspond to liquid handling. The other steps shown in blue are the signal collection for the detection reagent, which is performed by a microplate reader. So for normalization, we can say that liquid handling is the major contributor to the workflow. And secondly, it would also be beneficial for this workflow if we could also place samples into a reader. The other thing that stands out here is the transferring of samples from one type of vessel, for example tubes, into plates, which makes sample tracking very important. And this can greatly benefit from an automated tracking system. So if we automate all of the liquid handling steps in the normalization workflow, and if we include a plate reading in the automated workflow, then this bottleneck can now move on to another area. So now, with the bottleneck and its individual steps identified, I'd like to show you how Agilent addresses this bottleneck. So we would use the capabilities of the Encore multispan liquid handler to achieve a greater portion of the NGS workflow, and I'll explain to you why the Encore multispan is suited to this. So the Encore multispan has the capability to address many of the bottlenecks across the entire NGS workflow, from extraction right the way through to sequencing. With its onboard robotic arm, which can reach on and off the pipetting deck to move microplates and tip boxes and tube racks, allows the integration of devices such as plate readers or sealers, and a large number of other devices required for a particular workflow. New devices can be easily removed or added to the side or underneath the instrument. Another unique feature of the Encore multispan are the two banks of pipetters that allow for independent spanning across 2 dimensions. This allows us to address more samples across a wider area of the pipetting space at a single time. And also with room for 32 microplate positions, there's plenty space for a variety of accessories to be placed on the instrument, such as heating and cooling, shaking, and magnetic bead separation. Also, all the devices and accessories which can be integrated with an Encore multispan are also controlled by advanced scheduling software. So since this normalization application that we're using is an example, with pipetting being the predominant function, also the pipetter performance is very important for this application. So this is what I'd like to show you in this slide. So our instrument specification for the Encore multispan is for liquid transfers to be a 5% CV between transfers. In this graph, I'd like to share with you some of the CVs that we've measured with the instrument over a range from 0.3 microliters up to 200 microliters. So here I'm showing the CV for volume transfers using some of the different tip types on the vertical axis and the volume being transferred on the horizontal axis. Starting with a 10 microliter tip, pipetting DMSO, then using aqueous solution across a range of different tips. We can see that in all cases, which was measured across 8 replicates for each data point, the CV comes out to be below the 5% specification across all volumes tested, and the transfer volumes increase. As the transfer volumes increase, the CV continues to improve to where the majority of the volume range that would be transferred by a specific tip type now performs at a CV much lower than the 5%. I'd also point out that these tests were done using the industry standard RTEL verification system for liquid handlers. So the data is very robust. So with the performance of the robotic arm and the very precise pipetting of the multi-spanning pipetters, Agilent can put together a complete end-to-end solution for sample normalization, which also addresses many other steps in the NGS workflow. So in this slide, I'm showing the solution which would give the highest possible throughput for normalization. This workstation comprises of an Encore multispan at its heart, with a plate reader integrated to the left, a Bravo liquid handler on the top right for rapid full plate transfers, and a bulk dispenser in the front right for fast reagent dispensing. This workstation configuration provides the greatest throughput for a normalization process. However, many of the steps can also be handled by an Encore multispan on its own. The throughput would simply be different. For example, a lab could start with an Encore multispan on its own, then add some accessories and a plate reader integrated off to the side to give a complete walk-away solution, which would be able to process samples all the way from extraction through to normalization and library prep. By adding a thermocycler, the samples can be processed hands-off all the way through to loading on a sequencer. And as throughput continues to increase, a lab would start moving towards processing multiple microplates and eventually could benefit from doing full plate transfers to speed up the process. At this point, a Bravo could be coupled to the workstation. So as the needs of a lab grows, the automation could be expanded to achieve the necessary throughput. So with the workstation just shown, or a variation of it, I'd like to show how we create a complete walk-away workflow for genomic sample prep. So going back to my original diagram of the normalization workflow, I'm now showing in green which steps can be addressed by the Encore multispan-based workstation. And we can see everything can be covered in an automated fashion. I can start with unknown samples placed on the deck in either tubes or microplates, and then walk away and later be supplied with microplates of normalized nucleic acid, ready to move on to the next step in the sequencing workflow. The entire sample normalization is now completely walk away, and by adding more devices and some other accessories, a much larger portion of the overall workflow can be addressed. And now here in my final slide, I'd like to show you some of the data that we've collected by using an Encore multispan and a plate reader to normalize DNA samples. So here I'm starting with 96 samples of DNA at varying concentrations from about 1,000 nanograms per microliter down to 30 nanograms per microliter. Now this chart shows the concentration of DNA on the vertical axis and the sample number on the horizontal axis. So the starting concentrations, which vary greatly, are shown by the blue lines. In this experiment, we aimed for a final normalized concentration of 20 nanograms per microliter. The final verified concentration of the normalized samples are shown in the graph by the short red bars. It's hard to see the variation of the normalized readout just from this graph. So if I take the mean concentration across all 96 samples, I get 22.29 nanograms per microliter as the average. And the overall CV across these samples is 7.4%. Our manual comparisons had a CV of closer to 20%. So this data shows a great improvement over manual processing, and really shows the benefit of automating this process as not only increases the throughput and walk-away time, but also improves the overall data, not only in this step, but in the overall workflow downstream also. And with that, I'd like to thank you very much for your time. I will be here to answer any questions at the end, or please visit us on our website.
Louis, thank you so much for that detailed presentation on automating genomics workflows and on overcoming bottlenecks. You've perfectly set the stage for the presentations to follow. If you're just now joining our webinar, welcome. As I mentioned earlier, we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Daniel Tessier and Dr. Alexander Montpetit are set to begin their joint presentations. So, Daniel, you're up first.
Thank you very much, John. So my name is Daniel Tessier. I'm a Vice President Technology Centers at Genome Quebec, in charge of operations and business development for the McGill University and Genome Quebec Innovation Center. And I will be sharing this time presentation with my colleague Alexandre Montpetit, who is the Assistant Scientific Director at the McGill University and Genome Quebec Innovation Center. And he is in charge of the next-gen sequencing pipeline and will be addressing some of the technical aspects aspects of the presentation regarding automation and how we here at the Innovation Center are addressing quality and efficiency for providing better quality results for our users and for our internal researchers. So a brief summary of the presentation. I will be introducing who we are here at the McGill University Genome Quebec Innovation Center. I will be handing over the presentation then to Alexandre, who will be going through our next-gen sequencing pipeline and actually going through some of the protocols and some of the developments that we've made in terms of exome sequencing, capture, and automation, as well as our analysis pipeline, some project examples, and at the very end, I will be getting back on board to give you a little bit of a virtual tour of the Innovation Center, some of the pictures of the Innovation Center, and of course, acknowledging our partners and funders. So the McGill University Genome Quebec Innovation Center was inaugurated in 2002, just a little over 10 years ago. It is a partnership between McGill University and Genome Quebec. On the McGill side, under the scientific directorship of Mark Lathrop and a team of about 15 PIs, about 120 people altogether, in charge of research activities, various areas of genomics, proteomics, health, complex diseases, epigenetics, metagenomics as well. So a lot of research and technology development also done on the McGill side, as well as very strong bioinformatics and information technology infrastructure aspects, so IT. On the operations side, Genome Quebec with about 65 employees, averaging technical personnel as well as operations managers with individually more than an average of more than 5 years experience at Genome Quebec, essentially are in charge of the services that we provide and are also involved in tech development and tech implementation as well as bioinformatics and IT. And we essentially on an annual basis serve roughly 800 research teams and generate revenues of about $12 million in service revenues. So the mission of the Innovation Center is first and foremost excellence in science and in research. We provide advice and expertise as well as state-of-the-art services to our users. We implement technologies and applications and best practices in order to deliver the highest quality possible results to our users. And these users are either large-scale projects funded by Genome Canada, our mother organization, Genome Quebec, as well as the internal PIs, of course, of the Innovation Center, internal research, but also to the rest of the scientific community. We support health, forestry, environment, genomics in various technologies that I'll describe in the next slide, and we have a full access policy at internationally competitive pricing. And before I hand over to Alex, here's a very quick overview of the services that we offer here at the Innovation Center, starting with whole genome sequencing, exome sequencing, various custom as well as standard capture technologies. We have just recently obtained an Agilent certified service provider status, and Alex will go through some of the automation and examples of things that we've put in place. We also offer some RNA-seq, ChIP-seq, epigenomics, methylation analyses, transcriptomics, expression, low as well as high throughput sequencing and genotyping, some bioinformatics, and some data analysis, of course. So I will now hand over the presentation to Alexandre.
Thank you, Daniel. This slide shows a partial list of recent major projects using exome capture at the Innovation Center. I split the project in 3 categories, each usually requiring different amounts of coverage per sample. First, rare disease projects typically involve finding a causative mutation in a few samples. Examples include the FORGE initiative funded by Genome Canada and CIHR, which had the goal of finding new disease genes in pediatric samples, and for which I will tell you more about later. Other initiatives include Genome Canada Care for Rare and the Radical project at McGill, both of which having similar goals. The cancer projects we work on would typically require higher coverage due to the heterogeneous nature of the samples, while projects on complex traits would often involve sequencing hundreds to thousands of samples, but at a lower coverage, as we aim for common variants across the population. To be able to do all of these different projects, we needed a flexible platform that delivers high-quality data on a consistent basis and at the same time at a high throughput rate. Up to now, we've worked mostly with a SureSelect XT protocol, and in the past 2 years, we've done over 5,000 captures. Here are the various steps with their duration time in our exome capture pipeline. I will present each of them in the next slides and focus on those that are causing bottlenecks, which are mainly the library plus capture steps and the various data analysis steps. I will then explain how we address them. After DNA is sheared to fragments of about 200 base pairs using a covariance instrument, all the steps of the library prep and capture protocols have been automated. Up to now, we have used mainly the XT kit instead of XT2 because we feel we have more control over the sample pooling at the sequencing step rather than at the capture, in part due to the different target coverage we're using for our projects. In a manual mode, a technician can perform about 48 captures per week. Using a Bravo robot equipped with a 96-channel head, an orbital shaker, a thermal station, and a plate loader, we are able to increase the throughput to over 96 samples per week, and at the same time dramatically increase our reproducibility within or across batches. As such, we are currently getting a success rate of over 97%. To increase further our throughput, we started isolating the different steps and distributing them on multiple robots. For example, moving the numerous normalization and impure cleanup steps to a separate robot enables saving a lot of time overall, as it freed up the deck of the Bravo to do a second plate in parallel. For now, we have a throughput of about 144 samples per week and are actively working to increase it to 192 or even more. With the Illumina iSeq, we typically obtain about 40 gigabases of sequence per lane with 2x100 reads. We evaluated that to get good coverage of an exome, we need at least 10 gigabases, and thus we can multiplex 4 samples per lane. As indicated before, we would typically need more coverage when studying cancer exomes and would decrease the plexing. As each run lasts about 10 to 12 days, we are able to sequence the equivalent of 32 regular samples per week and per instrument. The size of the projects is usually not a factor, as we run 15 iSeqs at the center. But when we need a better turnaround time, we would sequence them using iSeq 2500 in a rapid mode and get data within 48 hours. Post-capture bottlenecks are thus seen primarily at the data analysis step. Our computing infrastructure is composed of several servers. Our main server, called Abacus, has over 400 terabytes of disk space for storing the data. It has over 1,000 nodes of 6 gigabytes of RAM each to perform the various analyses, which involve FASTQ read generation, genome alignment, and variant calling. However, for large studies, such as complex trait, this is not sufficient, and we move the analysis to a larger cluster with 14,000 nodes that is located on the McGill campus, and which is part of Compute Canada. Our pipeline for data analysis of exomes or genomes is depicted here. It includes tools for cleaning reads based on the quality and on the removal of adapters, tools for aligning reads to the genome and for evaluating the coverage, and then tools for calling variants. All of these tools are publicly available, and we have only integrated them in order to run the pipeline with as little human intervention as possible. Coverage is the metric we use to evaluate the quality of an exome and can be calculated in various ways. It can be defined using the targeted bases of the exome kit. However, we prefer using the CCDS or consensus coding sequences, which include only the well-annotated genes. Coverage, of course, is not uniform throughout the exome and vary between probes. The figure shows the distribution of the coverage of the first 3 quartiles of probes with the V4 kit. You can see that all probes within the lower quartile show less than 30x coverage. Some regions are constantly overrepresented, while some, such as those with over 70% GC content, have no coverage at all. Although we aim for an average coverage of over 100x, our threshold for a good exome is 60x. It represents approximately 80% of bases covered at 20x and 90% of bases covered at 10x. Variant calling can represent the most computer-intensive step. We use different tools and then report variant frequency from various databases, including AppMap and 1,000 genomes. Another tool we use is our own internal database that contains over 800 different exomes on the latest kit version. This is extremely useful in order to filter out various artifacts that constantly pop up in the data. End result is a much smaller file that would typically contain 200,000 variants per exome, and which could be displayed in a single Excel spreadsheet. You can use various tools to discover underlying disease-causing genes. One easy way is to use the list of variants and start by filtering for various parameters, such as the type of variant, and these would be, for example, non-synonymous variants, variants affecting the splice site, or frameshift mutation. You can then filter out variants that were seen in previously sequenced samples, as explained before, and then use a frequency of variants from the 1,000 genome database, as you would typically remove relatively common SNPs when studying rare Mendelian diseases. And then you will be left with only a handful of variants to prioritize for validation. One of the projects I mentioned at the beginning of my presentation, FORGE, which stands for Finding Rare Disease Genes in Canada, really turned out to be a success story. The goal of this initiative, led by Kim Boycott in Ottawa, was to quickly identify disease genes, that is within 2 years, from over 100 different Mendelian disorders. Disease-causing genes were identified and validated in over 2/3 of cases, which represents a much higher success rate than what is usually reported in similar initiatives. Some of the reasons include a tight set of rules for the inclusion of families in the study, and also great detective work by the scientists that analyzed the data, including Jacek Majewski and his team at the Innovation Center. All of this, however, would not have been possible without a generation of high-quality exomes done in a high-throughput mode.
Okay, thank you, Alexandre. In fact, these very impressive numbers with regards to the discoveries made through the FORGE project are really paving the way towards creating opportunities for clinical genomics applications. So very, very briefly, as Alexandre mentioned during his presentation, here's a very quick overview in terms of pictures. A picture is worth 1,000 words, so I'll stop talking very soon. But essentially, we have a platform of many sequencing instruments at the Innovation Center. And we have recently, as I mentioned earlier in the talk, been just recently certified as a Agilent certified service provider for target enrichment in 2013. But this certification actually adds on to many other certifications that we've had over the last 5 or 6 years with genotyping, expression, next-gen sequencing, as well as with a Center of Excellence for genotyping for various technologies. Just very quickly, again, a picture is worth 1,000 words. Just some of the users that we serve in 23 different countries. That was last year, 12-13, as well as various U.S. cities, both on the East Coast, West Coast, and mid-U.S. So in concluding Alex and I's presentation, I would like to thank our scientific partners as well as financial partners, primarily Genome Canada, Genome Quebec, CIHR, as well as McGill University. And with that, I will conclude and hand over the microphone to John.
Thank you, Daniel, Alexander, and Louis for those excellent presentations. If any of our webinar attendees were looking for ways to improve their genomics workflows, you've given them some great ideas and suggestions, so thank you again. We've had a number of excellent questions sent in by the audience for our panel for the Q&A session. But before we begin, I'd like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing soon. We would very much appreciate your feedback. As I said, we've had some wonderful questions from the audience, so let's take our first Q&A question. Okay, Louis, the first question is for you. What kind of DNA extraction techniques can you automate on the Encore?
So the platform is fairly flexible and open to most kinds of DNA extraction techniques. We can integrate a filtration station accessory onto the deck with a a vacuum pump and filtration and collar, which would work with SPE or filtration plates for cleanup, or else mag bead cleanup is something that we also do quite commonly with a magnetic bead plate and moving plates on and off of magnets, and sometimes using a shaker also to help resuspend the beads. So really, filtration or mag beads are the two most common ones we see.
And Louis, can your pipetter deal with blood clots?
Yes, the Encore Multispan has a liquid level detection system which is pressure-based. So there's a small pressure sensor at the top of the tip chamber that detects if there's a change in the pressure while the pistons are aspirating or dispensing. And that also functions to detect if a tip gets blocked as it's aspirating. So if there was a blood clot blocking a tip, this is detected by the software and alerts the user, or can try various methods for recovery or retrying to aspirate that step.
Okay, and Louis, what is the ideal reaction volume for the open tube ambient temperature incubations?
A lot of the volumes, I'm going to assume around the next-gen sequencing workflows, they tend to stay lower than about 200 microliters. There are quite a lot of temperature incubations we have there. Some of those actually benefit from sealing, and lidding can help quite a lot for extending incubation times. One of the other things that really helps extend incubation time if necessary is the tube or well geometry, which changes kind of the surface area of liquid for evaporation. But typically we do anything from 5 to 10 minutes down to as low as about 20 microliters without any problem, and then increasing the volume really only helps from there.
Okay, thank you very much. And Danielle, I have a question for you. Do you think the pipeline you described could be applied to a clinical setting?
Thank you, John. Yes, absolutely. Projects such as the Genome Canada CIHR Forge project and Care for Rare that Alex was mentioning have demonstrated the power of exome sequencing to identify variants directly associated with rare diseases with a phenomenally high success rate. And building on the power of this technology, its efficient management of pipelines, both technically as well as in a setting that would be accredited by whatever ISO or regulatory body, CLIA. Genome Quebec announced last week the establishment of a new clinical genomics laboratory dedicated specifically to the diagnostics, to more accurate diagnosis of pediatric diseases, and that will occur in partnership with researchers and clinicians at the Saint Justine Hospital in Montreal.
Thank you very much. And Alexander, what are the current bottlenecks in your pipeline?
Okay, well, our main bottlenecks are actually still located in the sample prep and in the data analysis steps. For the sample prep part, as explained earlier, we use different robots that each perform specific steps. So in addition to the Bravo that would do the end repair, ligation, hybridization, and other steps, we use a second robot, which could be also a Bravo, a smaller Bravo, but with a washing station, so that's pretty important, to perform the Ampere cleanup steps and also the normalization. And we use that second robot for the same task, but for other library prep workflows at the center. So we get different robots with different that have different specialties. So this way, you start to be able to advance 2 or even 3 plates at the same time, and using the same amount of technical personnel. And we're actually still evaluating closely ways to better organize the steps from our different workflows on the different robots so that we can increase the throughput and actually move the bottlenecks. It will move the bottleneck elsewhere. And for data analysis, we're still evaluating other existing tools with a goal to minimize computational time. Or we're also trying to find more efficient ways to better prioritize and distribute the jobs on the different servers. Also, we're considering other approaches like cloud computing also.
And Alexander, what made you choose the Agilent Bravo as your solution for automating library prep and capture?
Well, that's fairly easy. Actually, the Bravo comes with a complete solution for SureSelect or Alloplex protocols, and actually with a full support from Agilent. Also, what I can say is that we have that robot for the past 2 years. It has been pretty reliable, and we didn't have any major issue.
Thank you, Alexander. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Agilent, who made this event possible. Bye for now.